Bismillah Rahman Rahim. We start another paper. This is a paper 6.2 and it's an alternative to practical, which will now be a paper 4 in the new syllabus, which is for 2023. So this is also called ATP, or alternative to practical, which means it's going to investigate, it's going to check how well your practical skills are and how well do you understand the practical procedures that we do. Now, the paper that we're going to do is the November 22, and it is a paper 6.2. And this is, as you know, everybody knows this paper is for 1 hour, and this is called paper 6, alternative to practical. And then we know the marks for it is 40 marks, so total of 40 marks, and we need to be scoring well in this paper so as to get a good grade. Uh, Answer all the questions in the spaces provided. Yeast is a microorganism that can use the sugar sucrose to obtain energy for growth. When yeast is added to a sucrose solution, it grows, producing bubbles of a gas that cannot escape from this mixture, and so the volume of the mixture increases. A student investigated the effect of varying the concentration of sucrose solution on the growth of yeast by measuring this increase in volume. He started with a supply of 5% sucrose solution. He labeled 4 test tubes A, B, C, and D. A, B, C, and D. Right? State what you would use to label the test tube. Well, you use a marker pen or a wax pencil or a sticky label. That's all what you would use. He followed this procedure. Add 15 centimeter 5% sucrose to each test tube. So 15 centimeter of 5% sucrose. So this was 5% and the volume was 15 centimeter cube. 5% sucrose solution to test tube A. Then add 9 centimeter of 5% sucrose. So here we added 9 centimeter. In this we added 15 cm cube, and in this we added 9 cm cube of 5%, right? And 6 cm cube of distilled water. So 9 plus 6 we added distilled water. 6 distilled water we added. So 9 plus 6 is again equal to 15 cm cube. So 9 cm and 6 cm of distilled water to test tube B to produce 15 cm cube of, now this would be a 3% solution. So this is a 3% sucrose solution. This is a 15 cm cube of 5% sucrose solution, and here we have 15 cm cube of a 3% sucrose solution. And then you add 3 cm cube of 5% sucrose solution, and to this you added 12 centimeter of distilled water. So 3 cm cube plus 12 cm cube of water to test tube C to prepare again a total of 15 cm cube of 1% sucrose. So what do we have? What do we have here? I'm just going to clear this up and then show it to you again. So we have 5% sucrose. We have 15 cm cube of 5% sucrose, first one. The second one, we have 15 cm cube of 3% sucrose. So this was 5%, this was 3%, and then 15 cm cube of 1% sucrose. And then you added 15 cm of distilled water, and this was the D one. So we have A, B, C, and D. So this would be A, this would be B, this would be C, and then this would be D. He measured the volumes of 5% sucrose solution and distilled water with a small measuring cylinder. Describe how he could ensure that the dilutions produced were correct. So we have to find out how would they be correct. Part 2 of the question, how would they be correct? Well, it's very easy. How would they be correct? Well, if you measure the volume, if you measure the volume correctly, clean between sucrose and distilled water, rinse with sucrose solution. So we will actually ensure, because the question they've asked you, how we could ensure that the dilutions produced were correct. Now, this is what a measuring cylinder looks like. Measuring cylinder is something that we use to measure a very exact volume. So this is a measuring cylinder that we would use about, and let's talk about this now. So, I mean, just repeating the question, he measured the volumes of 5% sucrose and distilled water with a small measuring cylinder. Describe how we could ensure that the dilutions produced were correct. So you should clean the measuring cylinder between sucrose and distilled water. You use sucrose, then you wash it, then you put the distilled water. Then you rinse with sucrose or distilled water before the use. Then you use separate measuring cylinders for sucrose and water. So you have one measuring cylinder for sucrose and one measuring cylinder for water so that they don't contaminate each other. Then use a clean, dry measuring cylinder for each test tube, or you avoid the parallax, read the bottom of the meniscus and read it at eye level. So this would give you all the methods of how to use the measuring cylinder. So I haven't given you all the points which were clean between sucrose and distilled water, use separate measuring cylinders, use clean measuring cylinders for each test tube, and avoid the parallax and read the bottom of the meniscus. Now coming to the B part, he set up a beaker as a water bath at a temperature of 40 degrees Celsius. He added 1 gram of yeast to each test tube and stirred each mixture well. The 4 test tubes were then placed in the water bath. He decided that any change in height of the mixture in each test tube would be an indication of a change in its volume and so how much the yeast has grown. He measured the total increase in height of the mixtures in the test tubes in millimeter after 5 minutes and 10 minutes and recording them in his notebook. So after 5 minutes A was 40, 26, 25, and after 10 minutes 90, 64, 44 and 5. So he measured the total increase in height of the mixtures in the test tubes in millimeter after 5 minutes, 10 minutes and recording them in his notebook. Now it says complete the table using the information given. Now this is again for 4 marks. Let's do this. So this is the table. The table marks for 4, but what are the 4 marks for? Number 1, you have to do this heading, total increase in height slash millimeter. And then you just have to put these down, 5, 3, 1, 0, 40, 26, 20 and 5, and 90, 64, 44 and 5. So you just have to write them. So heading total increase in height was very important that you gave me this word total. And then heading units millimeter, this got you another mark for this 2 marks. Then sucrose concentrations entered correctly, this one got you 1 mark. And then all this data entered correctly got you 1 mark, both the columns. Use his results to describe the effect of sucrose concentration on the yeast growth. Well, what has happened? More sucrose, more growth. More growth, why? More respiration, more carbon dioxide produced. And less concentration, less growth. With 0%, there was with 0%. So the more growth between 5 and 10 minutes than between 0 and 5 minutes with sucrose, and little growth with 0% or in which there was no growth. So the points were, number 1 was this 1 mark, more sucrose, more growth. The second point was little growth with 0% or no sucrose, and more growth was between 5 and 10 minutes than between 0 and 5 minutes. So between 5 and 10 minutes there was more growth, and between 0 and 5 there was not so much growth. So there's a very good comparison which you should have brought out as soon as you saw this question. Then part 3, suggest an explanation for the results in test tube D. What was in test tube D? Test tube D had 0% sucrose, and it was 5, height was 5 minutes and then remained 5 minutes. So small amount of sucrose was present in the yeast cells and was already with the yeast or inside the yeast. So there was some which they didn't have any other outside sucrose, but it probably had some sucrose inside the cell, the yeast cell. A small amount of sucrose was present in the yeast cells. Part C of the question, he wanted to carry out this investigation at a temperature of 40 degrees Celsius. The thermometers in the diagram show the temperature of the water. And when he first placed the test tubes in the water bath E, and after 10 minutes F. So record the temperature when the test tubes were first placed in the water bath and record the temperature after 10 minutes. So E is what? E is 38.5 degrees Celsius. And record the temperature after 10 minutes. That is 27 degrees Celsius, right? So that was easy to check it. You see, this is 20, so this is 25, and then 26, 27. This is 35, and then 36, 37, and 30, between 30 and 40, this got to be, so 31, 32. This is between 30, so 35, 36, 37, 38.5, and degrees Celsius for both the temperatures. Now, suggest how the temperature of the yeast mixture could have been maintained at 40 degrees during this investigation. First of all, you use a thermometer. You would use a thermometer, and then at regular intervals over time continuously throughout, and maintain the temperature by adding hot water. Or you could have also said use a thermostatically controlled water bath. So using a thermometer, 1 mark, at regular intervals, you check the temperature so that you see if the temperature is becoming less, is becoming cold, so then you add a little bit of hot water. If it's becoming too hot, then you add a little bit of cold water, and you adjust the temperature by either adding hot or cold water, or basically they'd give you a temperature, and then they would say, okay, add a bit of cold, but they'd become cold after some time, then you'd add a little more hot water. Or you could have said a thermostatically controlled water bath, setting it at 40 degrees Celsius. So water bath at 40 degrees Celsius would get you a mark for that. Now coming to the last part of the question, D part, yeast uses sucrose to obtain energy for growth. Now if you look at the syllabus, this is respiration chapter, energy for growth. Suggest a simple method to demonstrate that the process required enzymes to be present in the yeast. Very easy. You heat the or you boil the yeast. You see, if you boil the yeast, what are you doing? Yeast is a living organism. If you boil the yeast, that you are denaturing all the enzymes. So you boil the yeast, this will denature all enzymes. And when it denatures all the enzymes, now what will happen? No respiration, no carbon dioxide produced, no bubbles, no increase in the height, or no change in the volume of the boiled yeast. So if still there was an increase in the height, then that means it wasn't the enzymes which were working, it was some other phenomena which did not require enzymes. Because as soon as you boiled it, then that means all the enzymes needed for respiration were denatured, they were deactivated, they didn't work anymore. So if they didn't work, means no respiration, no carbon dioxide, no bubbles, no increase in height. No respiration, so no carbon dioxide produced, so no increase in height. Now coming to question number 2, a student thought that seeds might germinate better in the dark than in the light. To investigate this, she set up 6 identical petri dishes, each with 20 seeds on moist filter paper. 3 of these dishes she placed in the dark and 3 under light in the laboratory. The lights were left on all the time. After each 24-hour period, she counted and recorded the total number of seeds in each dish that had germinated. She then calculated the mean number of seeds to the nearest whole number that had germinated in the light and that in the dark. Her results are shown in the table. Time, hours, seeds germinated. Now you can see here at 0 hours there was 0, 0, and then in the light at 24 hours, there were 0, then 11, then 13, then 15. In the dark, there was 0, then 5, then 15, then 16, then 17. So more have germinated in the dark, and they started germinating in the dark one. They germinated at in 24 hours, 5 had germinated, but 0 in the light. Construct line graphs of the data for seeds germinated in the light and in the dark using the same axis on the grid below. Join the points with straight lines. So now we have to do a graph of this. Let's look at it very carefully. As I've told you, the first and the most important thing is labeling the axis. Don't do anything else. First, label the axis. And the first column is going to come here on the x-axis. So that was time in hours. So this was going to come on the x-axis. As you can see here, we put it on the x-axis. And seeds germinated mean number were put per petri dish was put on the y-axis. So these are the dots for the graph that we are going to be making. And then we're going to connect them point to point with a ruler. So this would be the graph for this one. So the graph for this would be go like this, and then of course we would connect it with the, I might be slightly off at the points because you see it's not very easy for me to mark it, but I'm sure you all can figure it out. And then of course we would label this. Now, how did we mark this paper? If I was the paper checker, how would I mark this paper? Now the first thing is scale on the time on the x-axis and axis fully labeled with units. Got you 1 mark. Then linear scale for both axes over half of the grid used in both directions and at least 1 0 at the origin. And all points plotted correctly. Then straight lines rule to join plotted points correctly and no extrapolation. That means you don't extend it. It's only join the points that were given to you. And both lines labeled or a key used. So this will get you your 5 out of 5. Then coming to the part 2 of the question, use your graph to estimate the mean number of seeds that had germinated after 36 hours in the dark and show your working on the graph. So now this 2 marks is number 1 for working showing on the graph, and of course the answer was 10. Now let me show it to you, 36 hours in the dark. So if you see this point here, this is 36. 20 and 30 comes here, so 32, 34, 36. Each little small square is equal to 2, and then you draw a line there which goes up here, and then you see here it's 10. So you got your the answer was 10, and that is what you had to write. So you have to show me this line. This got you 1 mark. This line that you should have drawn in red, this line, you got you 1 mark, and then the fact that you wrote 10 here got you 1 mark. So line means show you're working on the graph. This is what was expected of you. Part 3, use the data and your graph to suggest what the student could conclude about the effect of light and dark on the germination of these seeds. Well, naturally, when you look at it, time, speed, rate, greater start of germination earlier in the dark, and number of seeds germinating being greater in the dark. So start of germination earlier in the dark, number of seeds germinating being greater in the dark. Now it says here ORA. Now what is this ORA abbreviation stands for? It stands for other reverse argument. Like if you talked about the light, that the number of seeds germinating being lesser in the light. So that was also acceptable if you gave me the reverse argument when they're comparing light and dark. So either you talk about the dark or you talk about the light. So if you give me the reverse argument, even that I would give you your marks for that. Now coming to B1, state 2 variables other than light that the student should have controlled in her investigation. And this is 2 marks. Always look at the marks first. Now this is water, temperature, and the same type or the same species or the same age of the seeds. And as you know, when I teach you this practical, I tell you TWO, the factors necessary for germination are temperature, water, and oxygen. So water and temperature could be 1 point. You could know there are 2 separate points. I'm not saying they're 1 point. They're 2 separate mark scheme points. So there were actually 3 mark scheme points. Same type of seeds, or the age of seeds was also correct. Same type of seeds or same age of seeds or the same species was also correct. Then suggest one reason for using 20 seeds in each of the petri dish in this investigation. Why were you using 20 seeds? Well, it becomes very reliable results. When you repeat, if you just used 1 seed and there was something wrong with that seed, maybe the tester was broken or maybe it had been eaten up by some fungus or some bacteria. So reliable results. The results are very reliable. If only 1 seed had been used and it didn't germinate, well, that was, but out of 10, if 9 germinated, well, then you know there were reliable results. And so then light is the only variable. Same number of seeds for light and dark allow a very good comparison. And some seeds might not be viable. Viable means they might not be living. Even they're in the dormant stage, but they are actually, because there is a time period for every seed. They remain dormant for a certain period, and then they can still germinate during that time, but then they will be dead or they will be damaged. So reliable results. So reliable results and same number of seeds for light and dark to allow a fair comparison. And some seeds might not be viable or maybe dead or maybe damaged. Now coming to the last question, the photomicrograph shows a simple plant that lives in ponds and lakes. G to H, there are two labels. The magnification is 630 times. In the space below, make a large drawing of the plant as it appears in the photomicrograph. So all you have to do is, it's very simple. All you have to do is you just have to trace it. One, two. Then look at the size of the nucleus inside. This one has a nucleus here, here. This one has it here, this one has it here, and then there's this one here. And then you have to draw this. So you draw it with double lines. Now, I've drawn it on this is just to show you this is exactly how you would draw it. So you would draw exactly what I've just said, but much bigger. It should occupy this whole space here. And you must look at this space given to you, and then you can easily, understand where the mark is. And so this should be somewhere like a big diagram here. Now, this might not be very exact because I don't have the picture in front of me and I'm just drawing it out of memory. So you just have to make it a very similar diagram to that, and it should occupy this whole space. You see, wherever you see the numbers here, so it should be nearly occupying 3/4 of this area. So big diagram, and now how are they going to give you? It's very easy, 5 marks. Everybody should have got these 5 marks. It's a very easy 5 marks. The marking scheme says 8 touching cells of similar shape drawn and height of each cell more than its width. Then sharp pencil continuous lines drawn for outline of the cells and no shading. At least G to H at least 70 millimeter. And nucleus in each cell and on correct side of the cell. You see, you have to see his correct side. You see, if you look at these, as I look at the diagram, it's very interesting. You see, these were here, one, this side, and this side. While these were on the other side. So this was how you had to draw them. So nucleus in each cell and on the correct side of the cell, and then two spines attached to top and bottom of each of the end cells drawn with double lines, and all four signs delimited from cell. All four spines delimited from cell. All the four spines must not have been a continuous of this. It should have been a delimited. Delimited means it wasn't part of the cell. It was separate from the cell. So delimited means having it separately. Because if you look at this diagram, see they sort of come out from the bottom of it and they come out like this. So they're coming out from some sort of the back and then they are coming like here. So delimited would mean that. Then B part of the question, draw a straight line on the photomicrograph to join lines G and H. Measure the length of this line and record it. So you have to measure G and H. Now this was about 42 millimeter. Always write in millimeters and that's what gets you 1 mark. So measurement 42 millimeters and then 42 divided by 630. Because 630, so you draw this line and you see this is 630 was the magnification, so you measure that 42 millimeter, 42 over 630, and this would be 0.067, 0.067, right? Describe how you would find out whether a sample of pond water contained this plant. 2 marks. What would you do? You would take a sample of the water and place it on a slide and then look at it with the help of a microscope. And you would actually have to use a high power lens because this is a very tiny high power lens, high power magnification or high power lens, and that is only then because it says how would you find whether a sample of pond water. So you take the sample of pond water, place it on a slide, add a cover slip to it, and then look at it under the microscope under high power, and you could probably see this structure in it. So that completes your this paper, and please do this paper before you come and watch this video. So best of luck and wishing you all the very best in your upcoming exams.